Well, thank you, Rick, and thanks to Findacure for the opportunity to speak to you today. I'm going to be talking in a little bit more detail about drug repurposing as a subject. And the first thing to say is that as a relatively new subject, the nomenclature around this is a little bit confusing. So for the purposes of simplicity, I'm going to assume that drug repurposing and drug repositioning and drug reprofiling, and even a term that I tried to introduce but wasn't particularly successful, but I tried anyway, and that was called therapeutic switching. All of these things I'm going to assume are the same thing. Obviously repurposing can include situations where you've got a generic drug which has been on the market for a few years, but it could also include situations where you're changing the indication of something which hasn't yet reached the market. And there are some other situations as well. So it's a complex field, but I'm going to assume that the nomenclature all operates under the umbrella of repurposing. I'm going to be talking mainly from the perspective of Healx, which is a technology company with a social mission focused on drug repurposing for rare diseases. But I'll also be talking about some examples of my prior experience at New Medicus, which was a consultancy I set up in 2008, specifically to advise people on the aspects of drug repurposing as a strategy for drug discovery. And the essential message of that strategy is that repurposing offers quicker, cheaper, and less risky R&D, and it's particularly useful in rare diseases. So as Rick pointed out, rare diseases actually, when you amalgamate them all, given how many thousands of them there are, as a body, they affect a large number of people, a population of roughly the equivalent of the size of a country, of the size of the United States. They're spread across 7,000 of rare diseases, and only 5% of them have an approved treatment. There has been an increasing recognition that repurposing offers a particularly attractive approach to rare diseases, and various funding pots of money have been allocated. In the NIH, they set up their first new division, which is called the National Centre for Advances in Translational Sciences, which has a focus on repositioning rare diseases. There's a program within the EU called eRare for rare disease repurposing. There's another organisation in the United States, which, if you like, is a sort of a sister to Findacure, although it's completely independent, called Cures Within Reach. And Findacure has strong links with them. It's founded by a chap called Bruce Bloom, and he set up a program within Cures Within Reach called Cure Accelerator. So it's becoming increasingly popular as a strategy for in rare R&D. And so let me talk about the particular particular sort of foundation stones for why repurposing is attractive. And the first is, I suspect, not unfamiliar to you, and that is that in pharmaceutical development, unfortunately, most journeys end unfruitfully. So this graph here shows the number of drugs that enter into the preclinical phase of pharmaceutical development, how many of them pass successively through phases 1, 2, and 3 of clinical development, and then they enter into registration, and ultimately they are approved. So for every drug that finally exits on the right-hand side of that graph, I would think, 14 have entered into phase 1 but not been successfully developed through to the end. And that money is effectively wasted. So the costs of R&D are magnified by all those failures, which ultimately don't lead to any commercial return. But even this is an over-optimistic view of the world, because what I've left out of that slide, I've titled it Pharmaceutical Development, what I've left out of the slide is the phase of pharmaceutical discovery that precedes pharmaceutical development, which is a vast kind of anonymous area which often takes place in universities, sometimes takes place in pharmaceutical companies, but in itself is a huge, expensive and time-consuming process. All of this leads through to huge costs in pharmaceutical R&D, and there are various ways of estimating this, and some of them quite contentious. But one simple way is to take the overall amount of money spent on R&D by pharmaceutical companies worldwide and divide it by the number of products which are actually approved by the regulatory authorities. And you can see that in the first place that starts off at a pretty high number. Back in 1996 or so, it's about $500 million. The number of new medical entities per $1 billion spent on R&D. So that's half of a product, whatever that means, half a product per $1 billion spent, and that's been getting worse. Between 1995 and 2005, the cost of R&D got significantly more expensive. But it may be that we've reached some kind of inflection in that process, but unfortunately, it's an inflection at a pretty high bar. So we're left with a situation where undoubtedly R&D costs an awful lot of money. The basic strategy, therefore, is to say, well, let's see whether we can do something with all those failures, whether we can, or even whether we can do more with the successes. And we can take the existing data we have on the drug that might have been available because it's on the market, or even something that's been partly developed but not fully developed. And as biology goes on, many of the discoveries which take place in academia, and we can combine the biology and the old data, and we can much more rapidly enter into clinical trial investigation. And this reduces the discovery time enormously. It reduces it from between 4 to 9 years to between 1 and 3.5 years. And also reduces risk, because the developmental probability of a product, of a de novo product that starts in clinical trials is there's about 90% chance it will fail, 10% chance it will succeed, whereas repurposing projects take out much of the safety risk, and therefore their chances of successful pharmaceutical development increase to about 25%. Despite the fact that the safety risk is much reduced, the efficacy risk, in other words, does the drug work in the new indication, that's mostly maintained, and that doesn't really surprise anybody, because one's dealing with a completely different new use. But if you put those two things together, the reduced time and the reduced risk, you can dramatically reduce the cost of new products. You can also theoretically obtain method of use patents, which is a great advantage, despite the fact, because Rick rightly says they are often successfully challenged. Sometimes they aren't successfully challenged, and sometimes they can underpin products with a 20-year patent term. And then finally, there's an additional opportunity in rare diseases, which I'll talk about in more detail in a few slides onwards. So, in summary, there are two radically different approaches to R&D. One on the top, repurposing discovery, taking a vastly shorter amount of time, 1 to 3.5 years in discovery, relative to conventional new chemical entity, or NCE discovery, which can take between 4 and 9 years. And then beyond those discovery phases, the clinical development phase is more or less the same. It takes between 2 and 7 years, depending upon the conditions, depending upon the trials you need to run, but ultimately ending up in regulatory approval. So there are a few take-home messages, in addition to those I've mentioned up till now, which might surprise you. The first is that most drugs have secondary uses. It's not a rare thing at all. Actually, most drugs are, if you take any individual drug, it is more likely than not it will have a secondary use. And that may be because there's the same mechanistic basis for the new use, or it may be because it's a different mechanistic base. Maybe because it works through a different enzyme or a different receptor then is the basis for the primary use. And therefore it's a vastly underexploited area. And a really important thing, which is often underappreciated, is that the dose, the route of administration, the duration of treatment and the safety can all be different, and these lead through to quite drastically different approaches in the secondary compared to the primary indication. As I said, it's still risky for efficacy failure and development. It would be hugely advantageous if we could define a commercial impetus for the drug in the new indication, because if we have the backing of a pharmaceutical company, whatever you think about pharmaceutical companies, they do provide a commercial underpinning for the commercialization of the product and the more widespread prescription and availability of the product to patients. And it's for patients, the purposes of patients, that I emphasize this point. Orphan drug exclusivity is one way of underpinning that commercial impetus, but it's not the only way. And then lastly, repurposing hypotheses can arise from a variety of different ways, and I'll talk about those in the future slides. But they could be computer predictions, or they can be, in some cases, retrospective evidence from clinical experience. So, the reason why we think that repurposing is more of a black swan rather than a white swan is because repurposing projects are often not commercially attractive and therefore not taken forward in the proportions or the numbers that are ideal. And without that impetus, the drug companies won't take it on. Without company investment, generally speaking, development will be slow, and or the quality of the data providing evidence of safety and efficacy will be poor. So let me give you an example of that. This is one of the oldest drugs we know. Acetyl salicylic acid is an extract of the willow bark known to Herodotus for the treatment of pain, now known as aspirin. It was one of the first pharmaceutical products marketed at the end of the 19th century for pain. And of course we know that it's not just useful for pain, it's also useful for thinning the blood. And it does so by disrupting the the aggregation of platelets. And that was discovered by the Nobel Prize winning pharmacologist Sir John Vane, just down the road in St Bart's Hospital. And there are products now on the market which are low-dose aspirin for the prevention of myocardial infarction and the prevention of stroke. What is less well known, but is also well evidenced, is that aspirin can be effective in preventing certain forms of cancer, particularly gastrointestinal cancers and other cancers. And this was shown from retrospective evidence. So this is patients who are taking aspirin, not for cancer, but for taking it for instance, it might be for pain, or it might be for prevention of stroke. And these authors from Oxford looked at 25,000 patients and found that there was a reduced chance of them developing gastrointestinal cancer, in particular pancreatic cancer, has a chance of 25% relative to the chance of you getting pancreatic cancer if you're not being administered a drug. So it's hugely protective for this form of cancer, yet there is no commercial impetus, because whatever I might want to do as a repurposing company to develop aspirin as a preventative treatment for pancreatic cancer, and I will have no commercial advantage and no opportunity to return the investment I make in that product, because any of you who want to benefit from that discovery can march down the road to Boots and you can buy whatever form of aspirin you want, not just the Medicus form or Healx form or whatever. And as a result of this discovery, it didn't actually originate from 2010. Obviously, it's been under investigation for many decades before. These things take a long time, and developing them takes a lot longer. So, many successful examples of repurposing, and some of them are quite remarkable. Thalidomide is, if you like, the byword for pharmaceutical poison. Yet it was reintroduced for the treatment of leprosy, almost as a Lazarus cure for somebody who had leprosy, and then subsequently found to be effective in the treatment of multiple myeloma. Sildenafil is, if you like, a poster child for repurposing, otherwise known as Viagra. What is less well known about Viagra is that it wasn't just repurposed once for the treatment of erectile dysfunction, it was repurposed twice for the treatment of pulmonary hypertension. Different dose, same product. Phinasteride is another interesting product which was discovered to effectively treat symptoms of maleness. By my maleness, what I mean is prostate hypertrophy and fullness. And there were two products actually introduced, both by Merck, one for the treatment of prostate hypertrophy and another for the treatment of male pattern baldness, again at a different dose. And lastly, glycopyrronium is a very old drug for the treatment of gastric ulcers, an oral product. About 15 years ago, a Cambridge-based company discovered that as an inhaled product, it had bronchodilating effects, and that product was then taken further as an inhaler. It is now on the market for the treatment of chronic obstructive pulmonary disease. And there are some other examples you can look at on the internet at that URL there. About 35 years ago, recognizing the difficulties that pertain to development of drugs for orphan conditions, the FDA introduced the Orphan Drug Act. And the Orphan Drug Act said that if a condition had a prevalence of fewer than 200,000 people, which given today's population in the United States equates to around about 6.25 per 10,000, then there would be various market exclusivities available to products that would treat that condition. And this particular legislation was extremely successful. Over 300 products have been approved since 1983, compared to fewer than 10 in the decade preceding the legislation. And as a result, the principle was replicated across most of the sophisticated regulatory authorities like the European Union, Japan, Australia, even Singapore. There were slightly different parameters as to what constituted a rare disease, slightly different lengths of exclusivity, but the principle was maintained. It was a partial solution, not a complete solution, because even with the orphan exclusivity, it's still legal and possible for a doctor to write a prescription for a generic alternative to an orphan drug, an orphan protected product, and that can give rise to cost savings to the NHS or to whatever provider is involved, but it can be to the detriment of the company that has been involved in the product development. So, misconceptions. Sometimes people think that repurposing doesn't identify groundbreaking drugs. This actually isn't true. There's a very interesting product called alemtuzumab, which was one of the first monoclonal antibodies prescribed for the treatment of chronic lymphocytic leukemia, which in a really amazing story was repurposed for the treatment of MS. And what it effectively does is reboot your immune system. So in certain patients, alemtuzumab actually offers protection against the progression of MS for up to 15 years. It's the most effective of any drug treatment for MS. Pirfenidone was originally for treatment of worms. It was then repurposed for idiopathic pulmonary fibrosis, which was a really serious and nasty condition. Ketamine, the street drug, but it's also been very interestingly proposed for the treatment of severe depression, and espindrol, which I'll talk about in a few minutes. I'm going to have to speed up a little bit because I've been told that we've only got a few minutes left. So what does Healx do? Well, Healx operates at the intersection of three things. One is patient charities, like Findacure, patient charities in the rare disease space. Secondly is the use of big data and transcriptomic data, transcriptomic information. And thirdly, in drug repositioning. There are three approaches we have to the identification of repositioned products. One of which is by comparing drug and disease profiles, gene expression profiles. Secondly is using retrospective analysis and finding unpredicted or non-predictive effects of drugs. And thirdly, we're developing some very interesting technology in predicting the effectiveness of drug, of which drug might work in a particular patient, which actually has quite strong connections with the principles of repositioning itself. The approach we take to using genomic data is to say that if you take a disease gene expression profile where you have an excess perhaps in a group of genes like that, then what we want to do with our drug is have an equal and opposite effect on that gene expression profile that's increased. So it's rather like looking at the profile of a mountain and a lake. If you see that deflection, then what we want to do with the drug is alter in an equal and opposite fashion the increased genes that are expressed in the disease. And actually there are a number of examples by people who've done similar things with similar technologies to ours and shown that this is to be quite an effective strategy for the identification of drugs that might work in particular conditions. And incidentally, it doesn't depend on a prior knowledge of the mechanism. It just says, this is what the gene expression profile is. We want to do something equal and opposite to it. So it's scalable and it's independent of the knowledge of the pathway. We've applied this in a number of rare conditions in collaboration with a number of charities, one of which is a very rare disease called NGLY1. It's a glycosylation disorder. And the second one is medulloblastomia, which is a rare condition, particularly affecting children, which is a cancer of the brainstem. We improve the accuracy of the predictions we can derive from the genomic predictions by adding in some literature mining technology and access to a database called drug repurposing.info. So with the combination of this real-world experience, we can improve the algorithmic predictions and identify repurposing opportunities which are accurate and insightful and in many cases protectable also, which can then be experimentally validated. I've got 2 or 3 minutes. I'd just like to quickly run through an example in retrospective evidence, because I think this is one of the most powerful methods of identifying repurposed projects. When patients are treated for any condition, the incidence of cancer is always recorded, because it's potentially the case that the drug itself is carcinogenic. But it doesn't always mean the drug is carcinogenic. It could mean that the drug actually is protective against cancer. But when you look retrospectively at the incidence of cancer, this is rather like the aspirin example. You know, the drugs can be protective as well as causing the condition. And actually quite a lot of drugs have been found to have protective effects in cancer. All of the drugs in the second column have been found to be effective against one or other of the subtypes of cancer on the left hand. Doesn't mean to say this is going to turn out to be effective prospectively, but it's a pretty good indication that the things are useful at the prescribed doses. It doesn't always have an impact in cancer. There are various other conditions where retrospective evidence has also been shown to have a predictive repurposing benefit. And I'd like to focus on cachexia. which is obviously a condition which accompanies cancer, as evidenced by that photograph on this slide of Steve Jobs prior to his death from pancreatic cancer in 2011. It's not just a chronic loss of weight, it's also a chronic loss of bone, nerve tissue, and functional muscle, like even the heart muscle is ultimately lost in cancer. And therefore it's an independent risk factor for mortality. Most patients, or many patients with cancer have it, and it leads to about a third of cancer deaths. So what some German cardiologists did was they said, we're interested in the potential for beta blockers being protected against cancer in heart failure. Now in heart failure, they actually use beta blockers for the treatment of heart failure, but you also get a certain incidence of cachexia, in other words, loss of weight over 60% within the period of 6 months or so. And if you analyse existing trials of beta blockers that have been investigated in heart failure, what they found was that beta blockers were protective against this weight loss. But beta blockers aren't all the same. We call them all the same because the name derived from a time before we realised they were pharmacologically heterogeneous. And to cut a long story short, what we did was we looked at all the beta blockers, which have a variety of pharmacological profiles, and we found that one of them actually had a significantly more effective benefit against the prevention of cancer than the others. And that one was a drug called Pindolol, which is a single isomer of an antihypertensive called Pindolol. And it even has superior effects relative to the racemate itself. So you can see it being about twice as effective as the racemate and reduces the chance of death by, this is in an animal model, by about 50% relative to placebo treatment. This was then taken forward into a phase 2 trial, a dose-ranging phase 2b trial looking at nearly 90 patients, high-dose Pindolol, placebo, and low-dose Pindolol. The primary endpoint was the rate of change in weight, and you can see at the bottom right-hand corner of that slide, the level of significance, even with this relatively small number of patients, is very, very high indeed. And you can see this graphically in the absolute weight change graph here, where the deep purple bars show the increase in weight as a result of Pindolol treatment relative to the loss of weight with placebo and low-dose drug. It's not just weight, it's also muscle. Muscle is important because it improves your functional capacity. It also protects you from heart attack. And again, you can see the protective effect of the drug. And you can also see an effect on hand grip strength, which is a measure of the functional day-to-day quality of life of the patients. Incidentally, there's no effect on blood pressure, which is always remarkable to me. So, in conclusion, I'd like to say that repurposing offers a radical way, a really radical way, of reducing cost, time, and risk in R&D, and it's particularly useful for rare diseases, which is why I'm talking to you here today. We don't just have to focus on the existing dose or the existing route of administration. These variables can be changed, and they can be changed to advantage in the secondary indication. Commercial incentives are important, they're not essential, but they're important to get companies involved, and the purpose of that is to be to the advantage of patients rather than to the companies themselves. Pindolol is an example where we took retrospective evidence and we progressed that through an animal model and into the clinic, which is currently the most effective developmental agent in this area, which has historically been a graveyard of pharmaceutical development. So with that, I'd like to conclude. Happy to take any questions, but thank you for your attention.